Hello, my name is Glenn Rubin. I am a PhD student co-advised by Dr. Bruce Clark and Dr. Ying Zhang, working on cool season turfgrass microbiomes and molecular detection of dollar spot, which will be the focus of this talk today. The objective of my research is to develop a molecular detection system to identify the dollar spot pathogen. Our goal is to identify and track the activity of the pathogen before visual symptoms appear. I am using a molecular technique called quantitative polymerase chain reaction, qPCR for short, to detect the pathogen in the leaves, thatch, or soil of the turf. The number of cycles required to reach the threshold indicates a concentration of the dollar spot pathogen. Cycle thresholds below 37 indicates that the pathogen is present, and lower values represent greater concentrations of the pathogen. I have evaluated creeping bentgrass cores that did or did not have visual symptoms in June and August. I separated each core into foliar, thatch, and soil layers and tested each for the amount of pathogen using the qPCR method. The results from the foliar tissue layer indicate that the dollar spot pathogen was present in every core, and as expected, diseased cores had a greater pathogen concentration than asymptomatic cores. The fact that I was able to detect the pathogen in asymptomatic cores indicates there is potential to track the pathogen in turf before symptoms appear. qPCR testing of the lower thatch and soil layers indicate that these layers are only effective for tracking the dollar spot pathogen once symptoms had already appeared. In summary, I have successfully developed a qPCR assay that identifies the presence of the dollar spot pathogen in field core samples before visual symptoms appear. Our results indicate that the pathogen is primarily found within the foliar and crown layers. This creates an excellent opportunity to study how the pathogen population develops in response to factors including turfgrass species and cultivar, fungicides, and environmental conditions. We are currently applying this detection method to better understand the spatial distribution of the pathogen in field plots. We are also seeking to understand the changes in pathogen population throughout the growing season, as well as the influence of cultivar and late-season fungicide applications. We greatly appreciate funding from the Rutgers Center for Turfgrass Science and the United States Golf Association to support this research, and thank you for your time.